Everybody, I am back with the second half of the IB Bio Chapter 9 Plants review. So we're going to talk about growth in plants, mostly hormones, and then also reproduction of plants. And I will try to make this go a little bit faster and more to the point than I did for the first one because it is almost 2 a.m. Okay, so growth in plants. Plants have these sections in them called meristems, and they're basically like sections of stem cells. Plants have these things called apical meristems, and apical meristems are at the stems and the roots. And basically, they can continue to grow throughout the lifetime of a plant. Some plants will also have lateral meristems, and lateral meristems, instead of helping them to grow up and down, they will help them to get wider. Okay, so when we're talking about the apical meristems and the plant growing, mitosis is going to play a big role in that because we're going to need to divide existing cells to make new cells. And every time a cell divides in the meristem, one of those daughter cells stays as a part of the meristem, and the other one is going to become a part of the mature plant. And obviously, root apical meristem is going to be responsible for the growth of the root, and shoot apical meristem is going to be responsible for pretty much everything that's not the root, so it'll be for the growth of the stem and for growth of leaves and growth of flowers. And like I said, every time the meristem divides, one of the meristem cells is going to stay as a part of a undifferentiated meristem, and the other one is going to become differentiated and become a leaf or a flower or a stem or a root. So what we're seeing here, this gray one over here, that is the shoot apical meristem, and these green things around it are supposed to be representing daughter cells from divisions, from previous divisions, which are then going to kind of branch off and then become like a developing leaf or something like that. This section here, I don't think you guys really need to worry about, so I'm going to continue along. Okay, so when we're talking about plant growth, you have to talk about plant hormones, which are then going to regulate growth. And the class of hormones that's really important in plants, they're called auxins. And basically, auxins initiate the growth of roots. They also influence the development of fruits, and they regulate leaf development. And one of the types of auxin that is very common is called indole 3-acetic acid, or IAA. And it's going to control the growth of the shoot apex. And it's going to make the stems become longer, and it is something that the apical meristem cells produce in them, and it gets transported down the stem to stimulate growth. So this is going to help with vertical growth. However, when there's lots of IAA auxin, then it can also inhibit growth. Okay, the next thing I want to talk about are these hormones called cytokinins. Cytokinins are hormones that are made in the root, and they are going to promote axillary bud growth. And axillary just means like additional bud growth or just budding of leaves off of the meristem. And the ratio of cytokinins that you have to auxins is going to determine whether or not the axillary or extra bud is going to develop. And then another type of hormone that is important to know about for plants are called gibberellins. And gibberellins are hormones that contribute to stem elongation. So pretty much as we're talking, we've got our auxins and our cytokinins and our gibberellins, and they all have to do with promoting growth. Auxins are going to be the broadest type that promote growth, so they cause root growth. They also cause fruit growth. They cause leaf development. And they also cause the growth of the shoot, or the thing that's going to become the stem. And then you've got gibberellins, which pretty much just have to do with stem elongation, and then cytokinins. And cytokinins are going to be more focused on growing the axillary buds. And axillary buds up here, they are going to be the shoots that form at the junctions or nodes of the stem and the base of the leaf. Okay, so moving along. We also need to know about plant tropisms, and basically it's how the plant knows in which direction to grow. And the two tropisms that you need to know are phototropism, which means plants grow towards light, and they do that because they need light for photosynthesis. And plants also respond to gravity, so it's called gravitropism, and that basically means that they grow into the ground and are going to root themselves into the ground. And one of the ways they're going to do that is through auxin. So auxin gets transported from cell to cell in the plant through these proteins called PIN3 proteins, right here. And basically where the auxin is where the plant is going to elongate. So in the case of phototropism, as we're looking at this picture, if the sun is right above the plant, then the PIN3 proteins are going to transport the auxin, or in this case, the specific type of auxin is IAA. It's going to transport the auxin to the very top of that plant. Because the sun is right over you, we're just going to grow towards the sun. However, if we see here the sun is on the right side of the plant, the PIN3 proteins are going to pump the auxin to the left side of the plant, and that's going to promote growth on the left side of the plant. And if there is more growth on the left side of the plant than the right side of the plant, that causes the left side of the plant to bend towards the sun. So that's our example of phototropism. And we also mentioned gravitropism. And it's the same idea. You're going to have shoots grow up and we're going to have roots grow down, and it just has to do with gravity. And so if a root is placed on its side like we see right here, the gravity is going to cause cell organelles called statoliths, which are just like these circles right here. The statoliths, they have the PIN3 transporter proteins that transport the auxin. And so if you have the root laying on its side, the organelles of the statoliths are going to transport the PIN3. They're going to transport the auxin using the PIN3 proteins that they have to the rules, or I guess according to the rules of gravity. So if the root is laying on its side, then we're also going to basically transport the auxin to this side using the PIN3 proteins inside the statolith organelles. And if you have a bunch of auxin on this side, what that should cause is that this side is going to elongate and cause the root to curl and then hopefully go into the ground towards gravity. And then once that happens, we see here the root is in and it's going into the ground. Then we have pretty much the same amount of auxin being transported to both sides of that root hair, and then it should grow equally on both sides. And then over here, if that's not the case, then it's going to have more auxin be transported to one side, and that should cause uneven growth, but the correct kind of growth so that the root digs into the ground and follows the rules of gravitropism. This orange box here you guys don't need to know about for Miss Cunningham's test, but it's fair game for paper 2, and it's just talking about micropropagation. And basically the idea is because plants have this section called the meristem that is able to kind of grow on and on and on and on, you can cut portions of plants as long as you keep the shoot apex or shoot meristem intact, and you can plant it in a piece of medium. So this is called agar. It's just a gel with nutrients, and this is called micropropagation. So then this plant, which was cut off of an adult plant, will be able to grow into its own adult plant, same as this one over here. So then you end up making these identical plants. So this is basically plant cloning. And if any of you guys have succulents at home, you can pretty much micro-propagate succulents. And this is just going off of the same thing, and it's not going to be on your test this week with Ms. Cunningham, but you may want to take a look at it before your IB exam. But if you didn't have time, then this is not one of those boxes that I really think you need to worry that much about. And I'm going to skip through that one too. And then we're going to talk really quickly about reproduction in plants. You guys know that flowering is kind of the most efficient and most celebrated way of plant reproduction. And what we mostly want to focus on in this chapter that's probably newer to you all is long day plants and short day plants. And they start to talk about it a little bit here, talking about temperature and how that is going to influence flowering, but long days and short days are kind of directly correlated to temperature. So I'll skip over that. And the same thing, long days and short days are directly correlated to the amount of light that you have. So the most important thing that you need to look at is this section right here that talks about photoperiods and flowering. So there are two kind of classes of plants. We have for flowering plants or angiosperms, you have long day plants and you have short day plants. Long day plants are going to flower when the days are long, which is in the summer, and the nights are short. Short day plants are going to flower when the days are short and the nights are long. So those make sense. It's not one of those weird things in biology where it's the opposite. So now we know that there's two different types of plants, long day plants and short day plants. The other thing that we also need to know is there is a pigment in the leaves of plants that kind of are able to measure the length of the day. And based on the length of the day, they are going to be able to transcribe the gene that allows them to flower, or they're not going to transcribe the gene that allows them to flower. So based on temperature and amount of light and basically what this pigment is noticing about the environment at that time of year, we're going to determine as a flower, do I flower? Or as a plant, sorry. As a plant, am I going to flower or am I not going to flower? So the pigment that we need to know about is called phytochrome, and it has two different forms. It can either be phytochrome R or it can be phytochrome FR. And there's a couple things that you need to understand about how this phytochrome works. When phytochrome R absorbs red light, it gets converted into phytochrome FR. Phytochrome FR, when it absorbs red light of a different wavelength, it gets converted back to phytochrome R. However, this left side of the equation doesn't happen as often as this right side of the equation because sunlight that we have on Earth is more of this wavelength of red light, which is 660 nanometers, and less of this wavelength of red light, which is 730 nanometers. So basically, phytochrome is just this molecule that absorbs light, and it switches between being phytochrome R and phytochrome FR. In kind of regular sunlight, we convert phytochrome R to phytochrome FR. Okay, so that's all we need to know so far. There's one more thing that we need to know. And the one more thing that we need to know is that phytochrome FR is able to convert to phytochrome R or PR slowly in the dark. So phytochrome R changes very rapidly in sunlight to phytochrome FR. And phytochrome FR changes very slowly to phytochrome R in the dark. So what we're seeing here is in long day plants, when there's a lot of sunlight, that means we're looking at this right side of the equation. When there's a lot of sunlight for long day plants, then we end up producing a lot of PFR or phytochrome FR. When we have lots of phytochrome FR, long day plants are able to transcribe the genes they need for flowering. The opposite of that is short day plants. With short day plants, there's not as much sunlight, there's a lot more darkness, so we end up having a lot more of the PFR converting into the PR. So there's not a lot of PFR for the short day plants. However, they work differently. The receptor that they have inhibits the transcription of the genes needed for flowering when PFR binds to it. So if you have PFR in short day plants, that inhibits flowering. In long day plants, if you do have PFR, that is going to promote flowering. So long day plants, they like to have a lot of PFR. Short day plants, they do not like to have a lot of PFR if they want to flower. So, like we said, when there's a lot of sunlight for long day plants, the PR gets converted into the PFR. And the PFR is going to cause transcription and translation of the genes needed for flowering. In short day plants, the PFR is going to convert into the PR. So we won't have a lot of PFR left over once this conversion occurs. But short day plants do not like to have PFR around. So when there's less PFR, because there's been so much time for the dark that the PFR has converted into the PR, then the short day plants are able to transcribe and translate the things necessary for them to flower. Okay, that's a good image to look at. That's probably one of the more important images in the plants chapter is this picture right here and understanding these bullets that you guys have on the side. Okay, the other things that you need to check out are this flower image because you should know the different parts of a flower that can be pollinated. I won't go over that because that's kind of middle school 9th grade bio. You should also be familiar with mutualism and why mutualism is important to flowers reproducing and plants reproducing, and the mutualistic relationship between plants and pollinators. And then you just want to be familiar with the idea of seed dispersal, but I won't go over that. And we could skip this green box. You also want to be familiar with the structure of seeds. You don't need to know it that well, but you do need to know these different parts, generally speaking, for both the IB exam and possibly Miss Cunningham's exam. You just want to know the structure of these seeds. And this is just talking about how to do an experiment on germination. You guys should read what it takes for a seed to germinate, but it's pretty much just water and darkness usually because it's got to be in the ground. And then also it's got to be warm enough. It's also got to have enough oxygen. And gibberellin will be in charge of germination. So remember, that's one of the hormones we talked about. We talked about auxin, specifically IAA. We talked about cytokinin, and we talked about gibberellins. So just take a look at this page as well, but it's not super important if you have a general basis of how plants work. And I think that pretty much takes us to the end of chapter 9. So again, the important things, you want to know the structure of the flower, the structure of the seed. You want to understand PR and PFR and how they convert into one another. And I think that's pretty much it for 9.3 and 9.4. Thanks for listening.